Good morning, everyone. Do you hear me? Yeah, good morning. Great. Sorry, I've just arrived. I see that we are above the 100 already, so this problem was fixed. It took me 4 or 5 days to adjust with the Zoom people, so I apologize for last time. That's why I couldn't do it instantly. We had the videos that are up there, so hopefully whoever did not manage to attend last time did see the videos, and hopefully everything is fine. I will start by saying that the quiz, sorry, I'm not sharing my screen. Let me open that up. Okay, so let me share my screen. Okay, so the quiz is going to be opened right after the class, or maybe even during the break. I'm going to make it available. It's just 5 questions for 5 marks. It's an open book. You don't need to study for that. It's not memorization or anything like that. And you will notice with me, as much as possible, I hate memorization. I have a very lousy memory, and I don't count on yours. So basically, I want you to read. It's going to be a little bit of covering some of the pre-readings, the 2 papers for the pre-readings from session 1 or lecture 1. And it's going to be having 1 question or something from the lecture itself, from the previous lecture, not this one. And maybe another question in which you need to open the form GKB or something like that in order to be able to answer the question. I just want to make sure that you know how to navigate, how to open the system up, things like that. I'm just with the quiz, I'm just touching bases. The quiz would be open for almost a whole day. So it's going to be opened from now until Wednesday, tomorrow, 5 p.m. Again, it's an open book. This gives you the chance to go read, answer. It's going to be very easy. I don't suggest that you do it. As you see, it's an open book and I'm not monitoring you. There's no point going and asking friends, getting the answer from them, because honestly, this is for your own benefit. You need to read those papers. You need to navigate this website. You have enough time to do that. The questions are easy. So it's a guaranteed 5 marks. You don't need to, go around it. Just go ahead and take the 5 marks, do the tasks I'm asking you to do. The assignment, I started to download some of the assignments yesterday. I didn't mark them yet, but I will start marking them. For this assignment, this is the second assignment. It was already in the slides of this time, but you will understand it more once we start looking into today's session. I believe another question that we had was about the participation marks. How do you get your participation marks? Okay, participation marks are supposed to be soft marks, and the word by soft marks, it's not like a right or wrong thing. It's more towards helping you get better marks. Okay, so so with participation marks, my expectations is that you attend the sessions. My expectations is that you participate even with the messages as a yes or no or stuff like that. Answer the questions, participate in whatever we're saying, help out in the discussions. There is another part of the participation that would relate, the assignments are group assignments, right? So towards the end, you might be asked to fill a survey assessing your colleagues, like a peer evaluation. So the peer evaluation also would be considered in the participation marks, and not making the peer evaluation, but being evaluated by your peers, because I want to make sure that all of us did participate in those assignments. You're not taking the grades of somebody who did the work because they had to do it before the deadline, and then you're just taking the marks for that. It's not fair for the other people too. So the peer evaluation and the contribution that you did in those assignments would be reflected in your participation marks as well. Okay, so any questions for me? We're all good? Am I missing something in the chat? same problem with the chat saying I'm not sure why this is happening. Okay, so great. Okay, so we're all in the same page right now. No further questions or anything. Great. Okay, so we can start with today's lecture, and we have 118 people today attending. So first of all, what about the previous assignment? Was it hard to find the scenarios? Any difficulties related to that? Anything at all? Where to stop? Okay, I don't get it, Michael. So what do you mean by where to stop? Okay, bit general and vague. Okay, let me check here. Okay, it's hard to know what you were looking for specifically. Okay, I got your question. Okay, let me open the previous lecture here, the assignment session 1. Okay, so let me share my screen and we'll go through it quickly. This is not the model answer, but the model answer would be after nice working in a group, so we could discuss potential options. Exactly. That was the whole aim of working in a group, because I know the questions are not difficult. You could actually simply answer it by yourself. So it was a 45 years white male patient appearing. So I gave the ethnicity, I gave the age, I gave the conditions. He would start on sertraline, so that's he's actually eligible for doing the test because he's starting antipsychotics from scratch, right? Like naive patient. He had the results from the CYP2C19, CYP2D6. We only studied CYP2C19 and 2D6. This is the things that we did talk about. We didn't discuss them in details. We're going to do that today. And we understand now that how we're looking at the alleles. So the first question was, patient was wondering if he should share those results with his insurance company. This relates directly to the ethics. So yes or no? Should he share it with the insurance company? No, of course he shouldn't be sharing it with the insurance company. Okay. And before applying to the life insurance policy. So even if they ask it for, and then we have to, insurance companies, no, they are not the enemy. I'm not saying they are the enemy. They are not. They are helping us out for sure. But this is not right because the data can be used against him, right? His data can be used against him, can deprive him of his own conditions because, you know, of the regulations and everything, right? And could lead to genetic discrimination. Exactly. So we need to use some terms in here. We need to identify that what is the GINA and what is the GENA, right? The regulations and the year, specifically the one that's in Canada, because we are in Canada, right? And we need to advise the patient accordingly. So those are the regulations. This is what you need to do. You have to watch out. Your genetic information should be very private to yourself. You know, you do a proper counseling to this patient. So, you know, as long as the major points are covered, we're good, okay? Patient asks, even if he can choose to, he shouldn't. Exactly, even if he can choose to, he should not do that. Why? Because he needs to understand how this information can affect him negatively if it is in the wrong hands, right? Patient asks if we can share this information with his physician, and should that affect his current therapy? So should he share it with his physician? What would be our recommendation about sharing with the physician? Exactly. It's also, as long as he trusts this physician, he's another healthcare provider, he's a decision making. Exactly. And supposedly, yes. And then basically, as long as the physician is aware of his condition, then basically, yes, he's going to give him better therapy, better therapeutic decisions. He would be on board with the pharmacist in taking those decisions and slight manipulations in his therapy, based on his genetic or possible drug disease interaction. Comment on that and advise on the patient accordingly. If there is any change in his current therapy, how would you communicate that with his physician? What are your references? So basically, our references most probably would be either the CPIC, the CP, or the Pharm GKB. So we're going to visit the Pharm GKB. I don't have it on top of my head right now, but we're going to visit the Pharm GKB. We're going to see whether the regulations of the sertraline relates to the 2C19 or to the 2D6, right? And then afterwards, we are going to check his alleles, the ones that I just given you, and identify whether this is ultra metabolizer, poor metabolizers, normal metabolizers, or intermediate metabolizer. And based on that, we're going to check the regulations and decide whether his current therapy needs to be changed or not. So you need to tell me that this medication is affected by those alleles, by this specific enzyme, and thus you need to tell me that he is ultra metabolizer or whatever. You need to be aware of what is the score of each enzyme, and we're going to discuss that today in details. And then how to communicate that with the physician. You need to write that in scientific wordings, right? So basically, you're going to send to the physician and saying, wants to BCE can help guide his therapy. Yes, exactly. Yes, the score, I'm going to talk about it today. You didn't study that last time, so it wouldn't be fair expecting you. Basically, each each allele has an activity score, like a phenotype score. So whether if it is fully functional, usually it's 1. If it is less functional, it goes to 0.5 or 0. If it is ultra functional, it can be 1.5 or 2. Okay, so this is the score, okay? And then eventually when we merge, because each of us has 2 forms of the allele, like 2 forms, like here, when you look up here. So it's 3 and 3. For example, if this is 0 and this is 0, so the total is 0. If this is 1 and this is 0, so the total is 0.5 or 0, depending what is the normal metabolizers or comparison, okay? So this would be the score or the phenotype or the activity score for those specific enzymes, okay? Again, you're not supposed to learn anything of that by heart, okay? Increase or decrease the dose sertraline to ensure therapeutic effect without side effects. Exactly, and this is how we want to correlate that to the physician, make him understand, and so on and so forth. Okay, so I guess this would cover. And what are your references? You have to mention that this would be, for example, the CPIC or the Dutch group guidelines of this year that we reached through the Pharm GKB, for example, on the day of October, whatever, because the guidelines do change from one day to the other. This is how you need to refer to whatever references that you have used. Because, for example, if the guidelines today was not to change therapy, but if it happens in 5 years or in 1 year or in 6 months that the therapy needs to be changed based on this condition, then you and somebody goes back to you and say, okay, so based on what did you base your judgment? If the guidelines are saying this, well, basically we have access to those specific guidelines dated 2018 or whatever through this website on this specific date. And this gives you the appropriate referencing mechanism when you're taking a decision or a therapeutic decision based on this set of data. And needless to remind everybody that we need to be using the most updated version of those decisions or guidelines, okay? Am I okay with everyone now? Everything's fine, clear? Okay, so those are the expectations. You're not supposed to be writing a lot. You're not supposed, as long as the keywords are there, as long as you're hitting the main points in whatever we're discussing, then I think we're good, okay? So we're good with this one. And we're gonna start our lecture for today. So we're talking about classical and non-classical pharmacogenetics, okay? And towards the end, towards the end of this lecture, you need to be familiar with the fundamentals of genetics. So today we're going to go back a little bit. I don't want to go deeper. I know some of you already took some genetic courses, and eventually I hope that those would be some sort of a like prerequisite for our courses as we go forward, because you need to understand what is the DNA structures, the RNA structures, and what some terms are splicing, the coding, things like that. So we're going to go above that quickly today, just to be all on the same page. So we are all have certain understanding of the things, and then we go from there. To become familiar with the early pharmacogenetic polymorphisms. So what are the different sorts of polymorphisms? We are focusing on the SNPs, like single nucleotide polymorphisms, but there are some other terms out there, right? And how would that affect the PK and the PD? Okay, to be introduced to the common transporters, CYP450 and the receptors alleles. So there are different alleles. We're not gonna go deep today into that, because you are going to be studying that in your PK courses, some of it in your PK courses. I know that the PK course is running side by side to our course, right? With Dr. Brock's course, right? Yes. So so basically, Dr. Brock's and Dr. Tony, both of them are going to be helping you with those portions, but we're going to just touch base on that, and that's why I'm not going to repeat myself a lot, because you're going to be studying that. The part with the cytochrome P450s was already studied with Dr. Al-Qadi in the previous year, right? So so this is how we're going to manage, but we're going to talk about what relates to the alleles difference and stuff like that in this specific course. Again, how would we take a decision for precision health based on all the information for all the patients? Case management. So we're going to take a case, and similar to that, we're going to take a case for the assignment this time. So the assignment this time is another case that we need to deal with therapeutically. Last time it was more ethically. That correlates more towards how the lecture was going. And those are the different references in here. Just to remind you that we're in the pharmacogenetics, we're looking at the variabilities in the DNA sequencing and how would this affect the PK and the PD of the drug delivery. Okay. Now, let's start by the DNA. Okay. So DNA is deoxyribonucleic acid. It's a polymer of nucleotides. So here, nucleotides like amino acids. Okay, and it has sugar and phosphate and one of 4 nitrogenous bases. Here we have 4. Here we have the adenine, the thymine, the guanine, and the cytosine. Here. So the adenine, the A, the T, the thymine, the guanine, and the cytosine. Okay. And in the RNA, the thymine, the equivalent one for it is the uracil. So those are the basic or the backbone amino or nitrogenous bases or amino acids, okay? So the DNA is formed chemically of those amino acids along with sugar and phosphate. It is found in a helical structure, right? In a thread-like structures located inside the nucleus, okay? In a form called chromosomes. So those helical structures in here, double helix, okay? Formed in form of chromosome, and the chromosomes is located inside the nucleus of the cell. Each one of us have 23 pairs, one from the mother and one from the father, 23 pairs of chromosomes. 22 pairs, we call them autosomes, and 1 pair is responsible for the sex chromosomes, but it's not just the sex chromosome or the gender chromosome. It also carries some other, like the set of chromosomes carry other traits as well. So 22 pairs of autosome and 1 pair of sex chromosome. Okay, now what is the function of the DNA? That's a good question. What is the function of the DNA? When we say, okay, your DNA, your genes, what is mainly the function of the DNA? Let's see, suggestions here. Okay, to code for proteins. Okay, I like the word to code for proteins. Exactly. You know, I know you're much younger than me, but long ago when we used to have photos, we used to have a film, like, and we call it the negative. Okay, so we get this film and it has all the pictures on it, and then basically we go to a lab, where they take this film and they make it out of it photos or the copies of the photos. So, and we call this, it's sort of the blueprint, okay? So basically this one codes for whatever photo. So if you want, so the genes, some of the parts of the genes are coding, okay? Coding genes. And coding genes means they code, or they are the blueprint that we use to print from them the different things. So we have receptors, we have transporters, we have enzymes, we have all other things. We know that those enzymes, those receptors, those transporters, they have a lifespan after which they are decayed and new ones are formed. How are the new ones formed? The new ones are formed through going back to the gene and decoding that into producing more of those proteins. And I don't know whether you have studied some of the inhibitions. There is competitive and there is non-competitive inhibition. If you, for example, some of the enzymes, I don't know whether, have you heard those terms yet, or you didn't reach this point yet? Okay, so there are the competitive and the non-competitive inhibition. In the non-competitive inhibition for some enzymes, some of the enzymes are basically ruined for life. No, it's done. So those enzymes or those receptors or whatever, they got the inhibition that they are not going to work again unless new enzymes or new receptors are formed. So for the whole lifespan of this enzyme somehow is not going to work again. So if this is in those scenarios, I got the non-competitive. So basically it's a permanent damage for this, whatever that this happens. And then based on that, we check how things are going, okay? So, okay, so 98% are non-coding. So our knowledge until now is that most of the DNA, we don't know the exact function of it. Most of the DNA, most parts of the DNA in terms of size, 98 or 99%, we call them non-coding, okay? And we don't know what exactly they are doing, but we assume that they are somehow helping in the regulations of the process where the genes are translated into the proteins. They are helping in the regulation of the coding parts, okay? Only we could manage to decode, or we could manage to realize, according to our current knowledge, 1 to 2% of the DNA, and we found that their job is coding. So they act like the blueprint that encodes for the proteins, whether enzymes, structural elements, signaling molecules, etc., etc. Okay? So the DNA have the promoter region. So basically, once the DNA polymerase finds the promoter, it sticks to it, and then it unwinds it at this specific area, and then it takes the exons, which are the coding parts, okay, and they are collected in here, okay, in a form of transcription, forming from the DNA, forming the equivalent messenger RNA, okay? Those messenger RNA, or the exons, are generally after which translated into the proteins, okay, or into the amino acid sequence. So this is the general, or the abbreviated condition. So the DNA is going to unwind in here. The coding parts are going to be collected. The uncoding parts are going to be removed. The coding parts are going to be translated into a messenger RNA, okay? And then they are going to move, and then this is going to be interpreted into protein. How is this going to happen? So the pre-messenger RNA molecule undergoes a process called splicing, okay? And splicing here means that we're getting rid of all the entrons, okay, and collecting only the exons. So this exon here, this exon here, this exon, and so on and so forth. The same strand of the DNA can actually give you different genes or different enzymes or different structural proteins, depending on the sort of splicing. So here, this splicing took all the exons. This splicing took less number of exons. So the same DNA strand can be interpreted into different messenger RNA depending on the splicing process, okay? So the splicing process here results in an encoding messenger RNA, okay? So again, one gene can encode multiple different proteins, and sometimes even those proteins have different functions, okay? So this is how it happens. Now, afterwards, the messenger RNA moves from the nucleus to the cytoplasm, okay, where it meets a structure called ribosomes. So the messenger RNA attached to the ribosomes, okay, and begin to translate the code, okay, here. So the messenger RNA, each 3 letters in here, each 3 amino acids form what we call a codon. Each 3 amino acids form what we call a codon. And each codon is eventually translated using, okay, transfer RNA, okay, and it's translated into the appropriate amino acids. So for example, if we want to have this here, if we have C, okay, so that's the C, and the, sorry, that's the G, okay, G, and then we have the C, okay, and then we have the U here, okay, so that's alanine. So alanine here, this coordinate, the alanine here, has the coordinate of the G, the U, the C, and the U. Okay, so the transfer RNA, okay, is attached to the ribosomes here and interprets or makes an anticodon, okay, interpreting that into amino acid. Let's take another one here. We have the A, and we have the U, and we have the A. And this is interpreted into this amino acid, okay? And so on and so forth, okay? Each codon in here is interpreted into the equivalent amino acid. If you notice, you will see that there are sometimes different codons that are translated to the same amino acid. There are sometimes different codons that could translate to the same amino acid. So the AUU, the AUC, and the AUA, the 3 of them interprets for the same amino acid. Here, the GUU and the GUC and the GUA interpret for the same amino acids. Okay, so this is how the things are going. So again, to say it simply, so we have the DNA splicing occurs. Okay, it unwinds, splicing occurs, messenger RNA is formed, messenger RNA is moved into the cytoplasm from the nucleus, goes to the cytoplasm, attached to the ribosomes, okay? It meets the transfer and RNA, which transfer each 3 amino acids, is 3 sequential amino acids, which are the codons, into the appropriate interpretation of amino acids. The build-up of those different amino acids results in a protein. And then we have something that is called stop codon. If you see here, stop codons. Okay? Stop codons is the UAA, the UAG, okay, and the UGA. Those are stop codons. So when the transfer RNA meets those stop codon, the proteins is finally done and it stops coding here and the protein is released. If we meet an early stop codon, then basically the protein is truncated. If we meet something here, a mutation that happens in any of those codes, then we get a different amino acid. Then possibly, if the different amino acid that happens here, the protein here is different. The protein formed here is different. And as such, the mutations occurs and the phenotype and the functionality of those proteins, they do differ, okay? Any questions at this point? We're good? If you want to understand more, if you want to take it sort of a game, this, I mean, we don't have time for that here. It's a short lecture, but you can visit this website and you can play around by doing different coding and anti-coding things. So you get the 3 codons and then this, and then interpret it to the appropriate amino acid, so on and so forth, until you form your own protein strand. Okay. So this is something to practice on to understand this mechanism fully. Now let's go to the application of this. Okay. So when we say we have genetic variants or genetic polymorphisms, what do we mean here? We mean we have different types. We have single nucleotide polymorphism, we have indels, we have variable number tandem repeats, we have copy number variants, we have haplotypes. Okay, haplotypes. I'm not very much keen into the real definitions or the exact definitions of each one of those for the purpose of this thing. but we're going to go through them so that when you read them, you understand. Again, this relates more into the basic genetics and the basic courses that we talk about. You're supposed to be studying those in deep details, but as I mentioned here, we're getting all up to the level where we can start working together from here. Okay, so the insertion and deletion, this is a genetic mutation caused by an insertion or a deletion of a segment of a DNA of an organism genome, okay? Small insertion or deletions ranging from 1 to 10,000 base pair, okay, in length. So we know that the DNA unwinds, right? So the DNA unwinds, okay? And then this thing here is copied, okay? When it winds back, there is an insertions and deletions that happens, okay? If the insertions and deletion occur from 1 to 10,000 base pair, this is a small insertion or deletion, okay? Now we have the copy variant or copy number variant. It's a type of structure variation where we have a stretch of DNA. So the DNA is duplicated in some people. So there is a stretching of the DNA. Relation of this to human diseases, they couldn't correlate it yet. It's not yet known, but it is there, and they identify it as sort of mutation or polymorphism or variant in the DNA, okay? There are some cancers that are associated with elevated copy numbers of particular genes, but this is just merely correlations. They couldn't say that if this happens, it has to happen in every single cancer patient yet. But there are some correlations trying to figure out that if you see those mutations a lot, there is a high possibility of this patient or those population getting those sort of cancers, okay? And this is, okay, so this is individual one from the mother and from the father. This is the sequence, okay? If we repeat the sequence here, you will see that this is duplication that happens in here. Individual 2, okay? Structure where you have a stretch of the DNA, which is duplicated in some people and sometimes even triplicated or quadruplicated in other people. So individual 3, he has 1 extra in here. Individual 6, double extra, and so on and so forth. So extension. Haplotypes, combination of alleles or a set of single nucleotide polymorphisms found on the same chromosome. So if we find different SNPs or single nucleotide polymorphism happening, okay, then basically, and they are repetitive on the same chromosomes, we call that haplotype. And sometimes they call them clusters, so because they happen all together, they happen at the same time. So this and this and this, they do change usually with each other, and this is what we call a haplotype or a cluster, and it usually occurs together, okay? So this is how they define it. The easiest to understand and the easiest to discuss, and this is what we're going to be using in this specific course, because we're not here to study deep genetics, but we're more studying the applications of those genetics onto therapy management and therapeutic decisions. We're going to discuss more into the SNP or the single nucleotide polymorphisms, and we said that those are polymorphisms that are persistent and occur consistently in at least 1 to 2% of the population. If this happens, that means that this polymorphism is actually real, it is repetitive, it is happening, and I have to take this amount seriously. And so basically, if I'm having an A, and then all of a sudden I'm having a G instead of an A, or I'm having a T instead of an A, okay? So in the RNA, okay, so here we go. So if one of those nucleotides, remember when we were discussing yes, here. Remember when we were discussing here the interpretation? Okay, imagine that if this C is G, okay, so G, okay, we have G, and then G, okay, and then U, okay. So instead of the alanine. Instead of the alanine, I got the gly. The gly here is the glycine. So instead of the alanine here, I will have the glycine. Imagine if this happened again here. Instead of the G, I have a C or a U. Okay, so it's going to be here A, U, okay, A, G, E, U, G, E, U, G here. So I'm going to get methicine instead of the arginine. Okay, and as such, the block here forming the protein is different. Whether this relates to functionality or not depends. Okay, depends on the phenotype and how they study it, etc., etc. And then eventually they define those alleles as score 1, score half, score 0, etc. The things that we discussed earlier this morning. Okay. Okay, so we know now that the SNPs exist in 1 or to 2% of the population. It relates to single nucleotide change or single base pair change. Okay, it can be in the coding or the non-coding region, right? So the SNP can happen in the coding or the non-coding region. Sometimes SNP occurs, okay, and a single base pair is changed. But despite this is happening, I result in the same amino acid. Why is this? Exactly, because the different codons code for similar amino acids. So if this happens, this is why we see what we see. So despite that we have a SNP, we still got the same amino acids and no changes did happen, despite it was in the coding area, everything was the same, okay? Different proteins that could be functional or non-functional, okay? The proteins that occur from the SNPs, okay, can be either functional or non-functional, depending on how hard it is. And as I mentioned, they study each allele with a phenotype of it, and then eventually they give it a score of functionality. And based on this score, they eventually divide that. And we're going to see that in the two examples that we're going to discuss today, the 2D6 and the 2C19. Okay. So we have, this is the wild type allele, and this is the polymorphic allele, okay? When polymorphisms occur, does this polymorphism occur in the protein coding region? If no, then it is in the non-coding, okay? That this polymorphism has a functional consequence, I'm sorry. If no, we call this a silent polymorphism. So this is a silent polymorphism, nothing happened. Yes, we had a SNP, was in the non-coding, it didn't have any functionality, efficacy, then it's silent. If it was in the coding, in the non-coding, and then yes, it did affect the functionality, this could be due to one of two reasons. Either it affected somehow the transcriptor, it could be in the promoter region. So it did affect where the enzyme identify, okay? Or it could affect the splicing. So it could affect the transcription or the splicing, and as such, it changes in the functionality of the enzyme produced or of the protein produced. If it was in the non-coding region and it did not affect functionality, we call it synonymous. So we call this synonymous, okay? If it did affect the functionality, we call it non-synonymous. And non-synonymous could be missense or nonsense. And, sorry. Missense means the polymorphisms changes amino acid composition. It's a total missense. For example, the amino acid compositions are different, and as such, the protein loses its functionality, okay? The nonsense induces premature stop. Remember, we saw something called stop codons, right? We have seen some stop codons. So if the stop codons come a little bit early, so after, for example, 3 amino acids, instead of having like 15 amino acids, if the protein consists of 15 amino acids, after the 3rd amino acids, we got the stop codon. So the body stopped producing this protein after 3 amino acids. This is called missense, nonsense, because it's a premature stop codon. Okay, so the polymorphisms could be silent, could relate to transcription or splicing, could be synonymous or non-synonymous, and the non-synonymous could be either missense or nonsense. So now if we hear those terminologies, we really need to understand what does this terminology reflect, okay, in terms of functionality, okay, and in terms of reason. Those are things I expect you to know, okay, because we went through them and we discussed them. Any question at this point? We should proceed. Okay, let's go on. Okay. The polymorphisms can occur to any protein produced. So it can occur to enzymes, to receptors, to transporters protein. And if it occurs to any of those three, it would affect our PK and our PD because it's actually affecting the activity. It's actually affecting the clearance. It's actually affecting the volume of distribution. It's affecting the concentrations of the body. It's affecting the elimination process. So it would hit us in different parts of the ADME, which is the PK, or at the receptor site and the receptor sensitivity and the receptor efficacy, and that's the PD or the pharmacodynamics portion. Okay? So the boost polymorphism, or those changes in the DNA sequences and variations in the DNA would eventually affect your PK and PD, and this is how we correlate the PGX to the PK and the PD. Okay? Now, 50 to 75% of the medications are substrates of those phase 1 metabolism enzymes that are the cytochrome P450. Okay, we have the 3E4, we have the 2C9, we have the 2D6, we have the 2C19. We have a lot of enzymes that correlates to elimination or hepatic clearance or phase 1 metabolisms of medication. Okay. 50% of the CPIC drug guidelines involve the 2C19 or the 2D6, and that's how the 2C19 and the 2D6 variants do affect a lot of medications, so as other enzymes. However, they have been drastically studied and they have been thoroughly studied, and as such, we have a lot of guidelines that relates to the alleles or the different alleles of those medications with those specific drugs. Now, let's put things in perspective. Did you start studying clearance yet or not yet? Okay, so great. This would make my life easier. Okay, so if we did that, let's discuss. Oh, okay, so before we discuss the clearance here, let's first of all, let's see, those are the different, the technology was a little bit a factor of us, or delaying us from identifying DNA genomes, from identifying things. Technology now is getting better. In community pharmacy now, we have a lot of alleles that can be found or can be detected using commercial kits. Okay, there are other questions that are still out there. How valid are those kits? What is the quality assurance towards those kits, etc., etc. But to the best of our knowledge and to the best of the technology that's available in the market, those are the alleles that you can actually find yourself by just doing a commercial kit. We have the 2D6, the 2C19, the 2C9, the 3E4, the 3E5 COMT. We have CellCoB, that's a transporter. We have VK-ROC-C. We have the UGT, that's phase 2. Okay. Okay. So we have a lot of things that you can actually identify. Well, in addition to those, I think newly introduced is the mu receptors, the OPRM1. Okay, there are some P-glycoproteins that you can figure out. So there are a lot of alleles that you can actually test in a patient using commercial kits. Okay, the commercial kits, and we're going to discuss that in details later, but the commercial kits prices range between approximately $200 up to $500 or $1,000. Okay, until now, they are still not covered by insurance companies. Some insurance companies, they found a way of covering it, but most of the third-party insurance companies do not cover this. The patient has to take the decision to pay this amount of money by themselves to get those results, and then to share it with whoever they think it is appropriate. But as far as the technology is out there, it's as convenient as finding it in a pharmacy or finding it online or finding it in some labs within Edmonton and within other parts of the world. Okay, now let's go back. We were talking about the PK, we were talking about the enzymes, alleles, and we're talking about the SNPs, and we're talking about how now to correlate those genetic things that happened to our PK and PD, and as such the clearance, and as such the concentrations inside the body, and as such to changing doses. Okay, this is where we are right now. How can we utilize this data and this technology into our therapy management, okay? Okay, so we're going to be introduced now to different definitions. Did we discuss those definitions last time? I can't remember. Did we talk about poor metabolizer, intermediate, extensive, ultra? Did we talk about that last time? Okay. We sort of, yeah, we did not discuss that totally. We sort of maybe scanned it or mentioned it along the talk, but we didn't discuss that thoroughly, okay? Now, I'm gonna start writing here. Okay, yeah, exactly, but not in depth. Okay. Okay, I'm gonna write here because it's, do you see it when I write here? If I write, see that? Yep. Let's see. Yes, okay. Okay, so I'm gonna make this one a little bit bigger here. Okay, let's see what is here. Okay, so we have studied that we have clearance, right, equal dose by AUC. Okay. This is the rule that we have studied in the PK. Clearance equal dose by AUC. Okay. So in order to get, okay, exactly. This is the simple rule. Okay. In order to get, if anything happens to the clearance here, if I'm giving the same dose, this would be reflected in the AUC right away, right? So when I say I'm having a poor metabolizer, if we assume that this drug is hepatically metabolized and through the cytochrome P450, and that's the major elimination factor happening to this drug, okay? One of the major elimination portions happening to this drug, let's say 80% metabolized through cytochrome P450, whatever X, okay? And I'm saying that this enzyme or this cytochrome P450 is a poor metabolizer. It's not functional. It's non-functional. What would happen to my clearance in here? The clearance would low metabolism, low clearance, right? So if I'm still giving the same dose, what would happen to the AUC? The AUC would increase. What would happen when the AUC increases inside the body? Yes, the AUC increase, more effect and more side effect. I might reach the minimum toxic concentration, the MTC. I might reach the MTC, and then I'm starting to seeing some toxicities and so on and so forth. Okay. Okay. What happens if I'm having an ultra metabolizer? What I'm having if I'm C. So if I have here low clearance, then I have high AUC, then I have side effects. Okay. If I have high clearance, what would happen? Low AUC. And as such, I would see I might not be within the MEC, which is the minimum effective concentration. And as such, I'm going to have no efficacy or poor efficacy of the medications, right? Okay. So in this scenario, just to understand. So in this scenario, if I'm dealing with a drug that is mainly metabolized through the hepatic metabolism, and this happens, this is the thing I'm going to see, okay? So in case of poor metabolizers, what are the expectations? How can I adjust this? How can I adjust this? Decrease the dose. So I have to decrease the dose for those population, okay? Or if I don't have enough, I would change totally to another drug that is not metabolized through this enzyme. So either I decrease the dose or totally move to another drug, move therapy to another drug that is not metabolized by this specific enzyme, which is a poor metabolizer. Okay, so either or. Okay, now in the second scenario where I have a high clearance, what are the expectations? How can I modify this? Increase the dose. I have to increase the dose. In some guidelines, you will see me tripling and doubling and even 4 times increasing the dose. So more dose, okay? So this is by sense. This is by common sense. As a pharmacist, these are my expectations. But as a pharmacist and as a healthcare provider, can I take those decisions all by myself? Can I just say, you know what, just decrease the dose 50% over here, just increase the dose 100% over here? Do I have enough safety? Exactly. I need evidence-based decisions. I need evidence-based decisions. And this is why we follow the guidelines. But while we follow the guidelines, we have to keep things in mind so that if we read things that doesn't make sense to us, okay, then those things are either typos, there is something wrong with this decision, there is something happening wrong. So it's good to understand the basics, but when it's time to take decisions, I have to get to evidence-based results or evidence-based research, more than one ethnicity that has been done on, more than one test, more than one experiment. And I have those specific guidelines telling me exactly what to do. What if I increase the dose to the double and eventually I get side effects as well, along with the effects that I'm expecting? And how bad are those side effects that could be, etc., etc. Okay. The second scenario, if I'm having the same thing, but it is for a pro drug. What if I'm dealing with a pro drug? What would happen here? Okay, if I have the low clearance, sorry, not low clearance. What if I'm dealing with a pro drug and I'm saying it's a poor metabolizer? What's going to happen? Exactly. I'm going to see less effect or no effect. Either less effect or no effect altogether. Okay. If I'm dealing with that and I'm seeing an ultra metabolizer, what's going to happen with a pro drug? With a pro drug, more effects, or basically side effects, because as if you're giving a pulse, all of a sudden you're expected to see, for example, X concentration, and all of a sudden you see 3X or 4X the concentration all of a sudden in the patient's system, because I'm having like the enzyme working to activate this drug is super working. It's 2 times or 3 times working, okay? So the toxicity or high effects or side effects would be instantaneous, okay? So it's good to know whether I'm dealing, it's always good to understand whether I'm dealing with a drug or pro-drug, whether I'm dealing with a poor metabolizer or an ultra metabolizer, and this can make my understanding what are the expectations, what should I expect from this happening to the patient based on this therapy, okay? And as such, I get a tendency of what do I need to do to change that, okay? So in this scenario, the action is vice versa. So in this scenario, in the poor metabolizers, I either change the therapy altogether, okay, move him to something that is not a pro-drug, or to another drug that is not metabolized by this specific enzyme. And in this scenario, I have to decrease the dose if possible. Depends on how the guidelines or the evidence-based guidelines out there are guiding me to do. Now, is that clear? We're going to stop here for a break, 10 minutes break, and then we're going to come back to continue the lecture. Okay. So I'll see you at 10:10. Okay. Okay, everyone, we're back. Okay. Okay, so briefly, we're going to go over what we discussed. We discussed the genetic formation of the proteins, the receptors, etc., and how the DNA acts as the blueprint or the coding thing for all our proteins, including the things that relates to the PK and the PD. Okay, we discussed the different sorts of polymorphisms, and then we started looking into polymorphisms within the enzymes. And now we're going to just discuss, so basically the enzymes, specifically the cytochrome P450s, are divided into poor metabolizers, where basically the extensive metabolizers or normal metabolizer, and this is the wild type, or the everything is perfect, or when the SNPs resulted in synonymous version, okay, or if we had silent polymorphism. So all those results in extensive metabolizer or normal metabolizers, where we have the genotype and the phenotypes are within the normal range, okay. We have the intermediate metabolizers, when one of the two alleles is not as functional as the normal metabolizer. So because like each one of us carry two forms, one from the mother and one from the father. If one of the two forms is not working as good, or if both forms are working less, this we call intermediate metabolizer. There is the poor metabolizers, when both forms are non-functional. So if the two alleles are non-functional, then this is called poor metabolizers, very slow metabolism, which would result in the things that we discussed, whether we're dealing with a drug or a prodrug, okay? And then we have the ultra-rapid metabolizers, and those when one or more, one of the two alleles or two of them are actually working above the normal levels. And you might see here, oh, come on, chat thing as usual. Don't see the chats. Okay, stop share. Okay. Okay, what does that mean? Okay. The ultra, in some categories, you will see that there is the rapid metabolizers and there are the ultra rapid metabolizers. So this category could be divided into two categories. They form the rapid and then the ultra rapid, or in some other categories, it will be just categorized into four. So you'll see them categorized into four categories or five categories based on whether they divide this ultra rapid and rapid, they divide them into two, or they combine them into one category, okay? Now let's start looking into an actual enzyme. So let's start by the CYP2D6, okay? So the CYP2D6 have, in the poor metabolizers, we have two inactive or null alleles. In the intermediate metabolizers, we have decreased functional alleles. In the extensive, we have two functional alleles, and in the ultra metabolizers, we have greater than two functional alleles, okay? Now, this is by definition, like by the genotype definition. Okay, what about the phenotype? When we talk about the functionality, we have a phenotype, not just by looking at the allele and saying, okay, this allele is not working. So how can we determine if this allele is not working? This is where we see this line, and this line here describes the phenotype. Let's describe that in details, but let me tell you that for the 2D6, there are over 70 variants that has been identified. Not the functionality of the 70 are not all determined. Some of them we have strong evidence for the functionality of which, others not. We don't get enough information about how those functionality are working or affecting our medications. Okay, so this has been determined using genotyping, okay, and phenotyping. In phenotyping, the 2D6 specific probe. What does a probe mean? What does a probe mean? when I say an enzyme probe, what does that mean? Yeah, it's the substrate of an enzyme or a substance that sort of solely dependent on this enzyme for its metabolism. So it's basically when it's a specific substrate, or this substrate is mainly metabolized by this enzyme. Okay, substrate of the enzyme. Okay, so we're looking at, okay, so the relative ratio between the concentration of the probe, the dextromethorphan, and its CYP2D6 specific metabolite, the dextrorphan, is obtained. Okay, so as the ratio of the drug to its metabolites increase, okay, that means that I have more concentration of the drug versus less concentration of the metabolites. That means I'm a poor metabolizer. And if we see that this ratio decrease, then I'm having extensive metabolizer. Let's look back here. So the dm by the dx is more than 0.3. So if the ratio of the dm by the dx is more than 0.3, that means I'm having a poor metabolizer because the concentration of the initial substrate is higher. If the concentration of the substrate to the metabolite is between the 0.03 and the 0.3, I'm dealing with an intermediate metabolizer. If it is between this ratio, I'm dealing with an extensive metabolizer. And if it is less than this ratio, then I'm dealing with an ultra rapid metabolizer. And this is why we say that this is by the phenotype and this is by the genotype. So this definition is describing the alleles. So this allele is functional, not functional, whatever. But this one here is actually describing the enzyme functionality. Okay. Now, this is the activity scores I have been discussing with you guys, okay? And the activity scores are based, or is a phenotype sort of description for the enzyme or for the allele, okay? So alleles 1, 3, 27, whatever, are known to be wild type or normal, show normal activity. And as such, the phenotype score or the activity score for them is 1. Okay, now those alleles in here have decreased activity, and their activity here is 0.5. Okay, now we have non-functional, variant, or mutant, or no activity, and those are 0. So those sorts of allele are 0. Okay, so let's go back to farm GKB if you want. Okay. It's very hard to deal with this thing when okay. Sorry, dealing with the thing with the open, it's hard. Here you go. I'm in the farm GKB. Okay, and I'm gonna write, for example, what is a codeine? Okay, and I'm going to go to the clinical guidelines annotation. Okay, here the CPIC describes here with the 2D6. I'm going to go inside. I want to reach the calculator. This is why I opened this here today. Okay, and I'm going to put any of those alleles that we were just discussing. Okay, so if we go back down here to this table. Okay, let's choose any of those alleles. Let's choose number 1. Number 1 is a normal activity, right? And let's combine that with number 9, which is an intermediate activity. And let's see what activity score would this enzyme show us. So we'll go to the calculator in here. We're going to choose number 1. And we're going to choose number 9. Okay. They say until now it's normal function, but they are not sure. Okay, instead of putting number 9, let's put number 3. Okay, so the activity score is above the 0 because one is active and one is inactive, and less than 1.25. So it's reduced morphine formation, and as such, it's an intermediate metabolizer. This patient has one active allele and one totally inactive allele. Okay, so they define them as an intermediate metabolizer. What if we put the two of them as 3? Now the activity score is definitely 0 because both of them are 0. Okay, and it's a poor metabolizer. What if we put 2 and 2? Okay, now this activity score is still, they call it normal morphine formation, but it is some sort of intermediate metabolizer. They don't have enough information upon that yet. And that's why those calculators are good. These are the activity scores that you're looking at, okay? Some activity scores are quite clear. Like, for example, if we go over here and we choose, for example, 17 and 17. Let's see how it works. So if we choose 17 and 17. Okay, so this is reduced morphine formation. 17 and 17 here, both fall on the decreased activity. Okay, so when they define it in here, they say that it's an intermediate metabolizer. Okay, so this is the activity scores I've been telling you about. Okay, so when you see the activity score is 1, that means it's a normal. When it's reduced functionality, it's 0.5. When it's not functional, it's 0. And sometimes when it's above normal, it would say 2 or 1.5. That means that we're within the ultra rapid or the rapid metabolism. Okay. And how to confirm this? By going to the PharmGKB, using some of those calculators, putting them in here, and then eventually the activity scores and most of the things are going to come out to you, and they're going to help you out taking your own decision. In addition, within the PharmGKB itself, they are going to give you the recommendation because here they are adapting the CPIC. If we go for other ones, maybe they don't have the calculator, or they do, I'm not quite sure. We can try. So if we take, for example, the Dutch, usually the Dutch doesn't carry the calculator. It's the CPIC that carries the calculator. Okay, but they are going to give you some updates about here, the 2018 updates. We're going to reach that eventually because we'll be talking about the codeine today. Okay. Okay, so these are tables that we would see eventually. Okay, the 2D6 is involved in the metabolism of so many medications. A lot of antidepressants, antiemetics, antipsychotics, beta blockers, opioids, analgesics, and other medications. So this is the 2D6 is involved drastically in the metabolism of all those medications. Today, we're going to be we're going to talk about the codeine. So if you want to discuss about the codeine, let me ask you a question. Is codeine a drug or a prodrug? Somebody knows about that? Codeine is a drug or a prodrug? Prodrug. Calvin says it's a prodrug. Okay. Sydney, Ty, okay, Rebecca, excellent. It's a prodrug. So what does it form inside our body? What does it form inside our morphine? Exactly. It forms morphine. Okay. Okay, so it's an opioid analgesic. Okay, it's a prodrug of the morphine. It's found naturally, but at low concentration, so usually sensitized in morphine. Okay, let's see what we have here. Okay, so if we look at this mechanism of action in here, so the codeine is metabolized by the CYP3A4, 10 to 15%, into norcodeine, and then by phase 2 metabolism into codeine 6-glucuronide by 5 to 15% by the 2D6 into morphine. And morphine is the active entity of this drug. So if anything happens to the 2D6 over here, the morphine is going to be changed. Okay. Now it's good to know that once the morphine is formed, it goes to a receptor called mu receptor, and also the mu receptor have different forms. The mu receptor goes under the OPRM1, something like that, the gene-wise, and and this defines where the receptors for the morphine. So we have two portions here that can affect the codeine. We have the enzyme and we have the receptor. Both of those can affect how the codeine eventually give you the activity or the effect that you can be seeing. And this is how polymorphisms or alleles kicks in and alters in the codeine activity and functionality, okay? And as such in its therapy management. Okay, in the CPIC as well, we have a good thing, which is those videos. Those videos, I encourage you to see them, because in those videos, usually they describe the, you know, they give you a briefing about this medication. So they speak about the mechanism of action, and then eventually you get to understand whether it's a drug or a prodrug from how the medication works and is metabolized into whether active entities or less active entities or more active entities, right? And then it touch bases on the enzymes responsible for that, and it also touch bases on the CPIC guidelines, major CPIC guidelines, and any precautions. So those videos are usually 5 minutes in time or something like that. I encourage you, whenever you visit this page, if you have the video, go ahead and read it, because it's going to save you some time. And towards the end of our course, I want you to be very comfortable dealing with the PharmGKB, dealing using the calculators from here, dealing with the printouts from the CPIC. So the CPIC guidelines come in forms of papers. Okay, so the papers coming from the CPIC guidelines, I want you to be very comfortable reading them around, referring to some of the things are found in the supplementary tables, not in the direct tables. So you will find those are the tables here discussing what needs to be done. And then there are some downloads here talking about supplements. So there are supplements here. So some of the information you might not find in the main paper, but you might find in the supplement tables. Okay, so some of the information you might be looking at, because it happens to me once I gave a question for the students, and I provided the paper, but then the data was found in the supplement, not in the main paper. Okay, and that's why I'm telling you, familiarize yourself with all those things. Just get familiar, okay, with the different perspectives. Okay. And as you see here, for example, we have October 2019 update. So there is a certain update that happens in October 2019, but the update couldn't be as a big publication with the whole changes in it. It could be only just a few sentences about the differences or what was updated. Okay, so you might need sometimes to go back for the older versions of the PDFs and print them out and check them. Okay, here, for example, based on the CPIC guidelines, so it will send you here, and you will find that the original guidelines was found in the 2014. Okay, so the original guideline was found in 2014. There is an update in October 2019, and that's why I'm telling you, if I put a question in the exam, you have to clarify which year, which guideline did I specify, where did I ask exactly about it. And same thing if you're giving information to a doctor, if you're giving information to myself, how did you get this information, which guideline, which year, and through which place on what date. So those things you need to identify when you're referencing whatever guidelines or whatever recommendation, therapeutic recommendation you're giving out. Okay. Let's figure out here. Okay. Okay, so the codeine is a prodrug. It's bioactivated to morphine, a strong opioid agonist by the hepatic cytochrome P450 2D6. The efficacy and safety of codeine are governed by the 2D6 activity. Polymorphisms are a major cause of the 2D6 variability. Okay, and we mentioned something else, which is what else could be vulnerable to polymorphism within the codeine life cycle or mechanism of action? The receptors. It's called mu receptor. Okay, the mu receptors also can affect the codeine activity. Okay, great. Okay, now those are the guidelines. Okay, by the CPIC. Okay, so you'll find ultra rapid metabolizers. Okay, increased formation of morphine. So as expected, when we have an ultra rapid metabolizer, that means that we have high clearance of the drug. The high clearance means more morphine or more stronger pulse of a morphine coming in. So the recommendation here is avoid codeine use for the potential of toxicity. The evidence on this recommendation is strong. So another thing that you need to look at, whether the evidence is moderate, whether the evidence is strong, in also taking the decision and recommending those decisions to other therapists, okay? So whether this evidence is strong or moderate or unspecified, right? So these are things you really need to look at, similar to the activity score. What is the evidence rate? Okay, alternatives that are not affected by 2D6, because basically if you get another prodrug that is activated by the 2D6, we didn't do anything, okay? So this would be including morphine or non-opioids analgesics. If they are eligible to take the NSAIDs, then they are better taking the NSAIDs, okay? Tramadol to a lesser extent, because when you look at the tramadol guidelines, they say for intermediate metabolizers and ultra metabolizers, you can try, and if it doesn't work, then you don't use it, okay? But tramadol is also metabolized by the 2D6, okay? Hydrocodone or oxycodone are good alternatives because the are not good alternatives. Sorry. Because they are also metabolized by the 2D6. Here, they are not good alternatives because also metabolized by the 2D6. So if I'm giving you a multiple choice, I'm putting you in a scenario, telling you, okay, what should we do now? We have the codeine, this patient has alleles X and Z, and you put X and Z on the calculator, and X and Z tends to be ultra rapid metabolizers. What should I do? And then I put among the options hydrocodone or oxycodone. You're not supposed to be choosing this. Why? Because they are also metabolized by the 2D6. Then you're not doing the patient any favor by moving him from the codeine to the hydrocodone or the oxycodone. Okay. The extensive metabolizers or the normal metabolizers, normal dose. Intermediate metabolizers use label recommended age or weight specific dosing. Okay, moderate, so there isn't a lot of information, and this you can see clearly when you look at the CPIC. When we put things on the calculator, you will notice that they are saying still under study, we don't have enough evidence, well, follow the normal pattern of metabolism, similar thing, okay? And here we need to monitor if I'm going to monitor use of tramadol for just want to move this around. Okay, for responses. Okay, poor metabolizers and greatly reduced morphine formation. So I have to avoid the use due to lack of efficacy. And the evidence here is strong. And I have to move again to either the morphine or NSAIDs. Tramadol is partially accepted because of tramadol guidelines and the evidence based on it. But tramadol is also metabolized by the 2D6. Okay? And other things like the hydrocodone or oxycodone are not to be used, needs to be avoided. Okay, is this clear in here? Any question about this part for the 2D6? Okay, now I want you to, I don't know why is this so big. Yep, here it is. Okay, I want you to open the codeine thing here. Your assignment basically is on the, sorry, just give me 1 minute. Can't reach out my, here you go. Okay. Okay, so here we are. So I want you to get familiar that when you deal with the things in here, you will see that we have different guidelines. We have the Canadian Pharmacogenomics Network, okay? We have the Clinical Pharmacogenetic Implementation Constraint, the CPIC, and we have the Dutch, okay? All of them relates their decisions or the guidelines based on the 2D6. Sometimes you will find some guidelines related to 2D6, some guidelines related to 2C9, some guidelines related to 2C19. Okay, so for example, they would, like the Dutch would say, okay, so there are some guidelines related to the 2D6 and the 2C19, but the CPIC only puts, the guidelines only relates to the 2D6, okay? So those are where things could be possible, okay? If they don't have enough evidence, or you will find some papers now in the literature discussing about some correlations that were found upon polymorphism of certain enzymes or certain receptors, etc., etc. But the evidence isn't strong enough for those entities, for those guidelines entities, to start putting out guidelines and recommending things for the patients, okay? So I want you to start familiarizing yourself with this website, with those information. I made sure to put 2 or 3 questions in the quizzes and few questions in the assignment. I want you to start familiarizing yourself on how to use this resource, those resources, okay? Okay, let's continue on this one here. Okay, so there is a good question here. Compare between the 3 guidelines, and it would be very interesting reading through the Dutch guidelines. What happens when an adult dose above 20 milligram every 6 hours is prescribed to an adult? A lot of times the Dutch, when they go a little bit deeper, specifying a specific dose not to be increased, okay? More than the CPIC. The CPIC would give you like a general guidelines and would usually tell you a 50% increase or decrease of the doses, and then at this point you have to visit something else like the e-therapeutics or some other references to figure out what are the normal doses for those patients, and based on those normal doses you divide that by half or whatever. Sometimes the Dutch guidelines do specify specific doses not to be increased or decreased based on the ultra metabolizer, extensive metabolizers, poor metabolizers, etc. Okay? We're going to move quickly to the CYP2C19. And here are the tables that similar to the CYP2D6, okay? The different alleles with the functionality of those alleles, okay? The suggested SNPs where the SNPs has occurred, okay? This is the genotype, okay? And this is the phenotype. So 1 and 1, that's normal metabolizers, 1 and 2, whatever. This is another table from the CPIC talking about different similar scenarios. What are the alleles and what is the equivalent metabolizing or phenotype activity for those alleles, okay? Those are the medications metabolized by the CYP2C19, okay? So we have the proton pump inhibitors, we have the antidepressants and others, okay? CYP2C19 is involved with very important medications. Dr. Tony next lecture is going to be discussing about that roughly. And he asked me to tell you that he's going to submit his slide lectures sometime during this weekend. And he has 2 pre-reading materials, and he's expecting you to do a lot of discussions during his lecture. Okay, so what's going to happen is you are going to divide yourself into 2 groups. Some of you are going to read paper 1 and some of you are going to read paper 2, okay? And then basically you're going to form a discussion, and then you're going to go over the warfarin guidelines and possibly the clopidogrel depending on the tool, okay? What I'm trying to tell you is there are a lot of people worldwide working on this. This is some, for example, something I've seen in one of the conferences I did attend, University of Florida, discussing a project that they have done in hospitals where they have inserted the similar to what we see in our crawl systems or in our pharmacy management systems. When we see a drug-drug interaction, a pop-up would appear to you and saying, you know what, you cannot put those 2 medications together. There is a possibility of a drug-drug interaction. What University of Florida has been suggesting, and I feel it's very useful if we even adapt it in here, not necessarily for this specific medication, but for all medications as well. The CYP2C19 here, a pop-up would appear to the prescriber and saying, for example, that there is a drug gene interaction with the Plavix or the clopidogrel. And this would result into this side effect. And with the suggested alternatives for that would be this or that medication, okay, with those specific doses. As such, the prescriber seeing this pop up would modify his decision and as such adjust it according to the drug gene interaction. This is what we eventually aim to see in our pharmacy operating mechanism or systems in the hospital systems. This is what eventually we want to see in addition to our drug-drug general pop-ups that comes to us, that we have embedded in there the drug gene interaction as well. That being said, in another conference that I did attend, a very good question was held out there. And they say there are other factors, not just the drug gene is. Okay? And this is good for us as pharmacists to think about those ideas in our head now that you're studying the PK side by side. Okay, what if two medications that are in competitive inhibition, both of them are substrates for a 2D6, and we're dealing for a 2D6 poor metabolizer. The first medication, which is strongly has strong affinity or has a stronger affinity to the enzyme, is actually going to feel the poor metabolizer effect because most of this drug is going to be metabolized. But in this scenario, or in this complicated scenario, the second medication actually is already in a competitive inhibition with the first medication, and it's already not much metabolized by the 2D6 as it should be. Now, the question here would be, would the drug gene interaction telling me to change this therapy totally for this medication be valid 100% in this scenario versus another patient who is solely taking this medication without the competitive inhibition that we see for this medication? So what I'm trying to tell you is drug gene interactions are part of your therapy management decisions, part of the bigger picture, part after checking the patient's kidney function, the patient's hepatic function, the metabolism, or what if the liver has already problems and already the amounts of enzymes in the body are not the same that they are? What would be the effect on the genes in here? Okay, what if I have a drug gene interaction in here? Should I actually take it just like that, or should I think about it in context of all the other things that are happening for the patient? So I always say pharmacogenomics is not one entity that you need to follow. It's another factor in the bigger equation of therapy management for the patient. It's a bigger equation that has a lot of things: drug-drug interactions, competitive inhibitions, patient factors, special population factors like kidney, like liver, like age, like weight, like smoking. Okay, and then comes the drug gene interactions. So as a pharmacist, I need to be aware how to utilize this information and how to integrate it among the other information that I'm having to reach out to my proper therapy management and proper therapy decisions. So it's not a report that I need to read and just interpret to the patient, or a report that can easily a doctor read and give out to the patient. It's a whole therapy management scenario that a pharmacist needs to be on top of it. And this technology is coming fast, it's coming soon. You need to be prepared for it when it's out in the market. Okay. Okay, so in this one, we're going to solve it as a question and answer. What should happen if I have voriconazole, okay, poor metabolizer or intermediate metabolizer? Now I want you to open up your PharmGKB, okay, and put voriconazole. And tell me what are the regulations for poor metabolizer or intermediate metabolizers or ultra metabolizers and rapid metabolizers. Once you reach out for the results, I need some of you to guide me how to reach the same answer, to show it to all the class. Somebody there already? Is it CPIC or Dutch? What do you see? So somebody guides me around. What should I do? Should I write voriconazole in here? What did you do, guys?
Yes, John here. Yes, we did. I put in voriconazole in the Form K.
Okay. Where should I move next, John?
I quickly went and pressed just clinical guideline annotations.
So clinical guideline annotation. So did you choose to go for the CPIC or the Dutch?
I chose CPIC.
Okay, so you went for the CPIC. Okay, this is the calculator. Did you use the calculator or how did you get to your results?
Down into the table where it.
Excellent, yes. So
different situations.
Exactly. So those are the different situations in here. So for the ultra rapid metabolizers, okay, and he gave examples for that. Okay, choose. So this is the therapy management that would be here. It's telling you to choose an alternative agent that is not dependent on the CYP2C19 metabolism, right? Okay, and what was the question? This is the ultra rapid or the rapid metabolizers. Okay, and for the rapid metabolizers, also choose an alternative. Okay, now what happens for the poor metabolizers? Choose an alternative agent that is not dependent on the CYP2C19. Okay, higher dose adjusted trough concentrations of voriconazole. The second question is, now that we know the general guidelines from the CPIC, now if we know that the voriconazole is used to treat the invasive fungal infections in cancer patients, so this is a cancer setting, cancer patient setting, okay, and voriconazole here is used as preventive or it is used for the treatment of invasive fungal infection. And the trough, in order for it to be successful doing that, the trough concentration should be less than 2 microgram per milliliter. If the trough concentration is less than 2 microgram per milliliter, this is associated with clinical outcomes that are bad for the patient. So the patient might not die of the cancer therapy, but they might be dying of a simple fungal infection because we are not giving them an appropriate therapy. So if a patient is taking CYP2C19, 117 or 23, okay, now you need to go and visit where? Somebody else. Somebody guides me around. So I'm going to go back in here. And what should I do now? Somebody else, please.
I just looked at the table and I can see like allele 2-3 is poor metabolizer, and the implication says you'll have a higher trough concentrations if you have poor metabolizer.
Okay, so you use the table and it's saying that the 2-3 here is the poor metabolizers and you need to change the voriconazole into something else. Okay, which is perfect. That's a way to do it. What another way that is easier to do that?
Hi, this is Jessica here.
Yes.
The way that I did it is I just went up and then there's like alleles you can put in. So I put in like 1 and then I put in 17 for the other option.
Okay, so 1. Okay. Sorry. So 1 and 17 for the second option?
Yeah.
Okay, and it came as?
Yeah, it tells me that it's a rapid metabolizer, and then there's recommendations in terms of like choosing an alternative agent and stuff like that.
Exactly, and which agents exactly do they specify in here, whether it's the posaconazole, whether it is the isovuconazole or whatever, that is not a substrate of this specific enzyme, okay? So despite this looks easy because it's simply antifungal. So you would think, why would I care so much about the antifungal? But here the antifungal is used in a cancer patient setting, which is they are using a lot of medication to decrease immunity. They are using a lot of other like complicated therapy. The patient needs to have a trough level more than 2 microgram per milliliter, and they measure that actually through TDMs every once in a while. Okay. Now, this is a table I got from one of the conference. They are comparing the steady state plasma trough levels, okay, and for genotype 117 and 1717. And as you see here, this is the 2 that we should be here. So all the other genotypes, they are above the 2 for the trough level, okay. The 117, it's above the 2, but the 1717 falls here drastically below the 2. Okay, this is a study that they have performed and they have proven, okay, for the voriconazole. Now, they are comparing also the prevalence of the subtherapeutic trough percentage for the CYP2C19 ultra metabolizers, and the prevalence is more than 51%, whereas compared to the other phenotypes is less than that. Okay, why is this important? Again, this is not guidelines. You cannot use, this is like in terms of references, this is some sort of how to say that, this is research. Okay, so research is good. It gives you one evidence based on the setting of this research, on the population that they have recruited for this research. But as you go further for the CPIC, and they depend on this research along with other research. But this was good to prove the importance of those things, because in hospitals and in other settings, and when you attend those conferences, you will feel that those researchers, when they discuss those things, or those clinicians, when they discuss those things, they discuss it in terms of trying to implement doing those tests for those specific population, and trying to prove enough evidence for the people financing or taking the financial decisions for purchasing the kits, for purchasing the tests, for covering the tests for those patients. And by doing such studies, they want to prove or make evidence proof that the implementation of the drug-gene interaction for those specific population, despite it's just for the voriconazole, is very important, because it's going to prevent some side effects that would result in complications of therapy, in more hospitalization time, etc., etc., etc. So when those clinicians do those tests, they are not just mimicking other population or something like that. They are trying to prove evidence for the decision-making people that those tests are important, they are cost-effective. When we do those tests, even if we pay the $200 for each patient, we are still saving the patient life. We are still saving the hospital time and the hospital spots for other patients that need therapy, because we are preventing. We're working as preventive. We're preventing this and this and that. Okay, so those tests and those studies, when you see, they are very respectful. And when you look at it from a clinician point of view or from a decision-making point of view, those are very important things to show and very important things to discuss. And despite it looks like, well, this is like ABC, well, it's not ABC. Those are, it's a big, I wouldn't say fight, but I would say it's a big challenge out there for people working in this field to try to prove the efficacy of that, to combat the cost effectiveness and to combat the problem of the still expensive technology out there. Okay. This is the table that John and his colleagues out there did discuss with us. Okay, and that's it for today. This is the assignment for today. I'm going to make the quiz valid. I'm going to just highlight it, and I'm going to upload this lecture, okay, on this system as well. Any questions for me? Anything that is not clear?
Yeah. Quick question. Sorry, Dr. Hamdi. Regarding the next lecture with Dr. Kang, you mentioned dividing the group into 2 to read.
He likes to do it alphabetically based on the last name of the students, right? So students with last name from A to something are going to read this paper, for example, paper E, and student last name from this to that are going to read that paper, okay? And then you're going to... Yeah,
sorry, he'll do that later on.
I'm gonna. He already sent me an email. I don't have it on top of my head, but I'm gonna share that with you in a message on the e-class. Sometimes later today or early tomorrow, along with the papers that you're requested to read for the lecture. Okay. And yeah, and so I had another question about the assignments last time. Whether it was supposed to be in ADAPT format or not? Like, if you can put it in ADAPT format, I would appreciate it. That would give you more hands-on on ADAPT format. So if you want to answer this as one big case on ADAPT format, that would be better this time. Last time, it's not important. I didn't mention that, and I didn't push into doing that. But if you want to answer this as a case in ADAPT format this time, that would be even better. Even if we want to do it one on, one off, that would be still okay. But I mean, I need you to have hands-on on those things. Again, my thing is not to make your life harder. I want you to just have hands-on. I want you to run discussions. I want you to get more familiarized with, or more familiar with, all the things that we're going through and the ideas and the concepts we're putting on the table. Okay, so the quiz is going to be out right now. The assignment is out there already. It's in the slides. Same deadlines like last time. So Monday, November 2nd. Okay, and I'm going to upload what Dr. Tony is suggesting for you to read for next time. Any question for me? Anything at all? And thank you, Jared, for your help last time. It made a big difference. You saved all of us. Okay, thank you all, and I will see you maybe not next week, maybe the week after. Okay, if you have any questions, send me an email. Sometimes I'm not available, but I try to answer as soon as possible. Okay, and yeah, if you need an appointment with me, send me an email and we can specify an appointment. Unfortunately, I won't be available for a quick phone call and asking for a few questions here and there. Okay, thank you all, and we'll see you soon. Take care.